this webinar aimed at introducing to Mexican and Panama scientific community Human Technopole, Italy's new research institute for life sciences. My name is Emilia Giorgetti. I am the scientific attaché in the Italian embassy in Mexico City. And just a few words to present the event that was organized by the Italian embassies in Mexico and in Panama in the framework of the Italian Research Day 2022, and in collaboration, first of all, with Human Technopole and with the Institute of Biotechnology of the National Autonomous University of Mexico, which is celebrating this year 40 years of life and research in science. And Sinvestav, and in Panama, Senacit, which is the Ministry of Science and Technology. We will start with a very short welcome session with some distinguished guests. And the first one who will give us some welcome words is our ambassador in Mexico, Mr. Luigi De Chiara.
Thank you very much, Emilia. Buenos dias in Mexico City and Panama City. Buon pomeriggio, buonasera in Italia. It's a real pleasure for me to inaugurate this important seminar. First of all, I would like to thank all the institutions that are participating in the seminar. The Human Technopole in Italy, the Biotechnological Institute of the UNAM, one of the greatest and most prestigious public universities in Latin America. From Mexico, we also have Sinvestav, which is a net of public research institutions and laboratories. With both institutions in the past few years, thanks to the institution of a post of scientific attaché at the embassy Italian embassy in Mexico City, we have started and consolidated a very happy history of collaboration, and we look forward to increase this collaboration in the future. From Panama, we have the Senacit, the Ministry of Research, and the Gorgas Institute, which is the cooperation in Panama has just started, but looks really promising. I would also like to welcome all participants, also on behalf of Fabrizio Nicoletti, the Italian ambassador in Panama, who unfortunately cannot be with us today. But from Panama, from the Italian embassy, there is Giulio Mignacca, his deputy, that will say a few words as a conclusion of the seminar. A special thank to Human Technopole in Milan, and in particular to Dr. Jan Mataj, the Director General. It's a pretty recent initiative of research in Italy, but it's still consolidating its structure, but it has already become pretty operational. We are organizing this seminar in the framework of the 5th celebration of the Italian of the Day of the Italian Research in the World. Recently, the collaboration, the bilateral collaboration in the sector of science and technology has become a very important and defining moment of Italy's bilateral relations with the international community. And this year, the ministry has chosen the theme of life sciences. Well, first of all, because we are still fighting against the COVID pandemic, and for the past 2 years, health and life sciences have been at the center of the international collaboration. But also because it gives us the opportunity to introduce to Mexican and Panama authorities the new initiative of the Human Technopole in Milan. We have with us the director, Jan Matthai, and other representative of the Human Technopole, which will illustrate us the features of these new center in Italy, also with a view to explore possible ways of collaboration and cooperation. I would also like to remember that here in Mexico, we have a pretty robust channel of collaboration with Mexican institutions, and that exactly this year we are starting also to expand this collaboration to the health sector, and for the first time we have allotted some financial resources to allow bilateral projects in health and life sciences. I'll stop here, and without further ado, I hope that this seminar undoubtedly will be very interesting, but I also hope that will bring good fruits for the scientific collaboration between Italy and both Mexico and Panama. Thank you very much.
Thank you, Ambassador. And now it's a pleasure to introduce Dr. Eduardo Ortega Barria, who is the National Secretary of Senacit in Panama. It was a pleasure to meet you in Panama and a pleasure to meet you again here. Please.
Emilia, thank you very much. It's a pleasure for me to participate in this meeting. I will share very a small number of slides because I think it's a great opportunity to present what we are doing in Panama and what potentially could be a fruitful collaboration in the future. Let me try to do this. One moment, please, that I have some issues here with my presentation. 1 second. Give me 1 second. I have 3 screens and none of the 3 is showing the presentation. Let me do something here. Let me try to get out of here.
You have to share the screen. Martín, hay que compartir pantalla.
Yeah. I will. Let me try to do that. Okay, here. Okay. We will share number 2. Okay, let me see. Let me know, please, if you can see this, the screen.
No.
Not yet. Okay. To share. Let me see. Let me go to this side then. Sorry. Doesn't allow me to go to the screen that I want to share.
¿Está autorizado compartir, Martín? O...
Let me go here and see. I am open system preference, security and privacy to grant access. I think I need access probably.
Martín, hay que darle
Acceso.
la autorización a compartir pantalla, por favor.
Yeah.
Lleno, lleno.
Okay, perfect. I think that I can do it now. I am the co-host now.
Ya está como co-host.
Excelente.
Sí.
Déjame ver si. Let me close here. I promise you that I will try to use only the time that I was share screen. I will go to number 3 and share.
No, me sigue tirando.
No. Okay.
Se lo mando a Martín.
Yeah, please. We will send the presentation to Martin, but I can probably only share with you some of the topics that we wanted to discuss with you without any slides. Science is relatively new in Panama when you talk about owning our own agenda for science. Open system preference doesn't allow me, sorry. Okay, doesn't allow me to share the slide, but let me talk about it. SENACID is the National Secretariat for Science, Technology and Innovation, and is an autonomous government institution. The mission of SENACID is to promote science and technology as a tool for sustainable development and potentiate the scientific and technological development of Panama. We are responsible of articulating efforts to strengthen the science, technology, and innovation system by doing several things. One of them is supporting human capital development and programs related to STEM education, strengthening the capacity to develop research in Panama by funding all fields of sciences and working towards increasing the number of scientists and also supporting the scientific productivity of our scientists. And also important, we promote innovation as a vehicle to achieve competitiveness in the country. We do all of this guided by the National Science and Technology and Innovation Strategic Plan that is completely aligned with the goals set forth by the government National Strategic Plan. Over the last 20 years, in collaboration with the system in Panama, SENACID has developed 10 public interest associations. One of them is dedicated to digital alphabetization. You will hear from INDICASAP today, from Dr. Ricardo Leonard, the work that INDICASAT is doing in biotechnology and biomedicine. We also have a metrology center dedicated to national measure standards. Recently, we have created a regional center for innovation in vaccines and biopharmaceutical. This is a national and international collaboration where the Gorgas Memorial Institute is an instrumental part of the collaboration. We have a logistic and innovation center that is a collaboration with Georgia Tech University in Panama. Recently, as recent as 2 days ago, we created a center for educational research. We have collaborations with national universities in Panama, in particular with the Technological University of Panama. We have recently created a center for research in information technology and telecommunications. And with the National University of Panama, we are working to create a center for research and production under control environment. Of course, we have to work in the humanities, and we have a center for public policy and political and social sciences. And we are developing a technological center in Coiba. Is the largest island of the Pacific, side from Mexico to Galapagos, where we will work in marine ecology and biodiversity. Of course, SENACID is a small part of the Panamanian National Science and Technology and Innovation System, where the academic sector, the public sector, and the private sector plays a very important role. And I have to tell you that 90% of the budget of SENACID is dedicated to investment in innovations and research, and 90% of that budget goes to the national science and technology system in the country. We have identified 6 priority areas: climate change, digital transformation, energy, health, the agroalimentary sector, and also logistics. And in each one of them, we have identified areas of priority. Of course, we are in the process of trying to exit the acute phase of the pandemic. We cannot say that we have the pandemic under control, because we have learned from the virus and the evolution of the virus that you can have temporal control of the pandemic, but you will have again new waves, and we are living the 5th wave of the pandemic, and we are planning to invest in health over the next few years. The plan that we have for the future is to strengthen the national university capacity for research and innovation. Panama historically has been doing research based in institutes like the Gorgas Institute for Health Sciences, INDICASAT, the Smithsonian Tropical Research Institute, the Innovation Institute for IDIAP for agroalimentary. But we believe that it's very important that over the next few years, we focus our investment to try to strengthen the national universities and the capacity of the universities to do research and innovation. We will prioritize research and innovation as a source of economic growth and social prosperity, and we plan to increase the investment in R&D to catalyze knowledge generation, innovation, and technology. And the idea is that over the next few years, we will work in improving research infrastructure to retain and attract the talent in the country. Basically, we expect that over the next few years, the investment in Panama in R&D related to the gross domestic product of the country will be increasing, and we will try to approach something close to 0.5% of the GDP invested in R&D in the country. Thank you very much. And I don't want to end without thanking to the Panama Italian Embassy, and in particular to Emilia Giorgetti, who I met a few weeks ago when she visited the country, and she planned to organize this activity. And thank you, Emilia, for coming through with this meeting, because I think that from this activity, we can have different collaboration with Mexico and with Italy. Thank you very much.
Thank you very much, Dr. Ortega, for being here with us today. And now, Dr. Brenda, my great friend, Dr. Brenda Valderrama, who represents the IBT today in its 40th birthday. Thank you, Brenda, for being here, and we listen to your words.
Thank you very much, Emilia. Thank you very much, Ambassador Secretary. We are very pleased to be here on behalf of Laura Palomares, the director of the Institute of Biotechnology. You will listen afterwards with Dr. Tonatiuh Ramirez about the facilities and the capacities of the Institute of Biotechnology. Now it only rests to me to thank the Italian Embassy, not only for this communication, but for all the effort they have made to make science more amenable to the young people, to bring scientific projects to better end, and also for having very good friends from Italy here in Mexico. So I won't keep the floor long. So thank you very much, and I will see you afterwards in the next panel.
Thank you, Brenda. And last but not least, Dr. José Mustre de León. Dr. Mustre is Sinvestav's director. Sinvestav is a great partner for us here in Mexico. We have a lot of collaboration thanks to his effort and Marta Espinosa's effort. So thank you again for being here, Dr. Mustre.
Thank you so much. It is a real pleasure to be part of this webinar. And we're really happy that this can take place overall in a work climate in which, because of recent events, sometimes global cooperation is questioned. And I think in the case of science, we really know that it is a key ingredient for success. And in that sense, we're grateful with Italy as a country. I think the presentation of this institute is going to be really important for us. And in the past, other efforts by Italy to increase this international cooperation and incorporate overall the presence of Latin American scientists has been important, like the International Center of Theoretical Physics in Trieste, or the synchrotron facility also in Trieste, have been hosting Latin American scientists for a long time. And actually the active cooperations that we have had in the past has been crucial to the development of important groups, at least here in Mexico and in my institution, for sure. And we really welcome the involvement of the Italian embassy, of course, the great effort of Dr. Giorgetti. And in our case, one very important landmark was that during the visit of President Sergio Mattarella in 2016, we were able to sign an agreement of cooperation with the CNR in Italy that is still working, and it is being really beneficial for Simbe staff. I think just hearing about this human technopole will open a new step into cooperation. And even though we have had in the area of life sciences an active cooperation, I think Simbe staff will can increase this presence in the life sciences quite a bit. And I think, as you will see in this event with my colleagues, Marta Espinosa, Guillermo Elizondo, and Selene Fernandez, you will get a flavor of all the things that we do in Simbe staff in the area of life sciences. Another advantage of the initiatives that the embassy in Mexico has had is that this setup actually allows not only the direct cooperation with Italy, but in this case, for example, possible avenues of cooperation with Panama, and of course, in the case of the Institute of Biotechnology, we have had this strong cooperation for a long time, but when we are part of even more integrated efforts, it strengthens that part. So I think it has a lot of advantages, and I thank you, the Italian Embassy in Mexico, and Dr. Giorgetti for this opportunity. Thank you very much, and we're very happy to be part of this.
Thank you, Dr. Mustre. And now, before going to the technical session, just a few words on the organization of the seminar. There will be a first session during which Human Technopole will introduce itself, and we welcome Dr. Jan Matthij, who is here with us just to give the main description of Human Technopole, and Dr. Vannini. In a second technical session, Panama and Mexican scientists will introduce their institutions and present some technical works, investigations they are developing at the moment. And then a final session where everybody can discuss and propose opportunities of collaboration, new means to activate relationships and collaborations for the future. So the chairperson of the first technical session is another great friend of mine, Dr. Marta Espinosa Cantellano, who is the planning secretary of Sinvestav. Please, Marta, it's your turn.
Thank you very much, Emilia. It is really a great honor to be moderating this first session, which is the one that will present Human Technopole to Mexico and Panama. I am greatly honored to be chosen to moderate this session, and I thank the Embassy of Italy in Mexico and the Ambassador De Chiara and Emilia Giorgetti. So I would like to very briefly introduce the long and important CV of Dr. Ian Matash. He was an undergraduate in the University of Edinburgh in Scotland, and then made some graduate studies at the University of Leeds in the UK. After several postdoctoral positions in very important institutions, finally starting from very early in 1985, he was collaborating with the European Molecular Biology Laboratory, EMBL, where he served as group leader, then as program coordinator, after that as a scientific director, and since 2005 as director general. And these positions have been granted because of his very big capacities in research, in understanding the research areas that he heads. He has functioned in several scientific societies. I will only name about a few: the Future and Emerging Technologies flagship science coordination group from Brussels. He was chair of the European Intergovernmental Scientific Research Organization Forum, chair of the advisory board of the Wellcome Trust Center for Gene Regulation and Expression, and several honors and awarded memberships are in his curriculum. Among those is the recipient of the Science and Society Award of the Italian Chemical Society, and a member of the trustees of the Darwin Trust, a charitable trust dedicated to supporting young scientists from developing countries. He has also major scientific interests that I will not get into the details right now, but have to do with ribonucleic particles and the processing of messenger RNA precursors. What we have been waiting for a long time now is to listen from Dr. Ian Matash, the introduction of Human Technopole. And it is my great pleasure to give you, sir, the floor.
Thank you very much, Marta. And I will start by sharing my screen. Can someone tell me if everything is working okay?
Perfect.
Yes, perfectly.
Good, excellent. So, as I said, thank you very much to you for the introduction. I'd like to also thank the Ambassador De Chiara, for his introduction to the event, and both ambassadors for their participation in the organization. And again, as many of you already have done, I'd really like to give an extra special thanks to Emilia Giorgetti, because she was very active in making sure that this would happen and in expanding this to include both Mexico and Panama and through the embassies. I have to say that since coming to Italy at the beginning of 2019, I have found that the scientific attaches to the various Italian embassies are a very, very valuable resource and really are very active and enthusiastic people who get things done. So I particularly like to thank Emilia. So my task is to introduce to you Human Technopole. As was already mentioned, this is an institution which is in its early days. We started when I arrived in 2019 in January. There were 14 people. We are now close to 300 people, so we are growing quickly. When I arrived, there were a few offices in the building you see on this slide, which is the Italian Pavilion or Palazzo Italia from the Expo 2015. Now, as you will see later in the slides, we have a number of labs. We have space for close to 500 people, and by the end of this year, we will have space for 500 people to work either in offices or in laboratory facilities at the Technopole. So, what are we doing? The mission of Human Technopole, or the missions, I should say, of Human Technopole, are to carry out frontier research in the area of human health and well-being. We have also been given the mission to set up and operate large-scale research infrastructures within interdisciplinary laboratories. The research infrastructures, and I'll come back to this, will be open to external users, and we're still discussing exactly how this process will work. But we're already setting up the facilities. We want to act as an open hub to support the development of the Italian life sciences research community and to internationalize these activities. And we'll be engaged also with industry through working together with industry and through, of course, technology transfer activities. So what does an open hub mean? As I said, we want to set up research infrastructures and services that will serve not only our scientists, but will serve the external community, both national and international. We want to share expertise, infrastructures, and methods. So we want to have an active visitor program. But above all, we want to be collaborative. Nationally and internationally, we want to be a collaborative institute. We want to work together with other scientists and to carry out ambitious projects, which is always easier to do if you work as part of collaborative efforts. We'll be engaged in training, and I'll say something about that later in the talk. And as I said, innovation through working on our own or with industry. So where are we today? 60% of the researchers that we have hired come from international institutions. So we are attracting many scientists back into Italy or to Italy for the first time. You will see when I show you a few of the people that some are Italians who have never previously worked in Italy, but have come back now to join Human Technopole, and others come from other countries to work in Italy. And this is obviously one of the reasons why Human Technopole was created. We have a very ambitious program for recruitment, which will involve us hiring one scientist per week for the next 3 years. And this has already been going on for the last couple of years. So we now are well over 100 scientists, having first had accommodation for scientists at the end of 2019, beginning of 2020, and at that time, only offices and not laboratories. We have already started to raise external funding, and our researchers thus far have raised 8.5 million euros in external grants. And we have created, or are finalizing 20,000 square meters of laboratories and offices, which is a substantial structure, as I'm sure many of you will be aware. So Human Technopole is an ambitious project. The expectation is we will be somewhere between 1,300 and 1,500 people at steady state when we reach steady state. But it's embedded in an even more ambitious project in the context of Lombardy, Milan, and Italy, which is to redevelop the Expo site, the site of the Expo 2016, as an innovation campus. So the campus is large. It's like a small city in terms of the area. And it is now called the Milano Innovation District, MIND. Some of the main tenants who have agreed to come, including the first three public institutions that agreed to come, have a focus similar to Human Technopole around human health. So there is a very large hospital, a research hospital called the Galeazzi Hospital. I'm afraid I can't use a pointer on this screen, but the Galeazzi Hospital, you can see, is the structure on the top left of the screen, following the arrow. You can see where Human Technopole is located, and on the right of Human Technopole, the purple colored buildings will be the science faculties of the Statale University of Milano, who will move to the site and who have already cleared the site for their buildings, and which will start to build their buildings very soon now. In addition, there are the plan is to bring innovative industries onto the campus. The first two tenants who have joined the campus, who have moved here, are AstraZeneca, one of the companies who created one of the successful anti-COVID vaccines, and Illumina, the genomics company, the DNA and RNA sequencing machinery company. And it's obvious that Human Technopole was a major attractor for these companies to come to the campus, and we hope they're the first of many who will join the campus, and that we really can create an innovative park. So going back now to Human Technopole, we, as I said, have begun to create space, and we have created roughly, or will have created by the end of this year, roughly 20,000 square meters of space, enough space for about 500 people. And we've done this through renovating some of the pavilions that were pavilions of the Expo. The building on the bottom left, labeled neurogenomics, genomics, and computational biology, is one of these two buildings. In the top of the screen at the center is a facility where Alessandro Vanini and his team have put a lot of effort into creating facilities for cryo-electron microscopy, for structural biology, and others have put effort into creating a light microscopy facility there. The Palazzo Italia building in the middle will be our administrative buildings, but also will house a large number of computational researchers, as well as the data center. And then finally, we have made use of prefabricated laboratories in order to quickly create space which surround what's called the tree of life, the structure on the bottom right. The white buildings surrounding this are all prefabricated laboratories, which are now up and running, and where the Neurogenomics Research Center, the Genomics Research Center, and the Structural Biology Research Center are all currently located. The green transparent block on the left of the screen is a building which is designed but not yet built. It will be finished probably sometime around the end of 2026. And this will be a building that will have space for roughly 1,000 people in it. And it will be mainly for researchers, so laboratories and offices again. And this is a picture of the building. It's an innovative building. As you can see, it has a green roof. It's very attractive and very open. And the idea is that the campus, like the entire Mind campus, should be open for people to walk around, obviously not inside the buildings, but on the outside of the buildings. It should be attractive to visit and to walk around. So what are we doing in these buildings? So the first task that I had on arriving was to decide what would be the initial focal areas of our research. And obviously, because of where science is, it was a very easy choice to have a strong commitment to genomic research. Genomics is making human beings very interesting experimental organisms. We can really do experiments using genomic technologies, using non-invasive technologies and non-disruptive technologies to gain knowledge about human beings, including to gain knowledge about disease states. So functional and population genomics is one of our research centers. And there's a second which is focused on neurogenomics, because neurodegenerative and neurodevelopmental diseases are among the most important unmet health needs that we're currently facing. Structural biology was a choice because there has been a revolution in structural biology methods. In other words, there has been a revolution in the use of cryo-EM in structural biology over the last perhaps 12 years now. And this technology is enabling many things that would have been impossible before. And Alessandro Fanini will introduce the structural biology research that we are doing and intend to do in the future immediately after my talk. There is a center called Health Data Science, but perhaps I go first to computational biology. All of these areas of science produce huge amounts of data. And so we need to have very extensive computational analysis methods, including people who are experts in artificial intelligence, machine learning, etc., to try to gain information from all of the data that we will produce. And in the Health Data Science Center, in other words, in the last research center, the intention is to use these big data methods to look at the health system itself and to look at aspects of the health system and to try, through making use of large data methods, to extract information that will be useful for policymakers in this area. And I've actually moved on unintentionally to the next slide, but those areas are all supported by large-scale research infrastructures. And some of those are essentially beginning to operate now. They take some time to set up, as you can imagine. Genomics is beginning to operate. Cryo-EM is beginning to operate. Some of the light microscopes are functional and are working. There is a very innovative center, which is an automated stem cell and organoid facility, which is attempting to automate the production of organoids and the manipulation of the cells that are used to make the organoids, such that the experiments can be done on a much larger scale than is currently possible. And then, as I mentioned, that we need a large data center to collect the data and also to share the data with the scientific community. So let me just show you a slide that has a lot of faces on it. So we have been recruiting in spite of COVID. We started to recruit in 2019, and we recruited 6 of the heads of research center, which are the people along the top line, and Andrea Sotoriva in the first year, and were able to do this in person. There were 3 group leaders who were also recruited during this period, so more junior researchers who will lead independent groups. But most of the other people on the slide, as well as most of the people in Human Technopole as a whole, have been recruited during the COVID period. In other words, we have been very active in recruiting through the use of video conferencing and through virtual interviews, with a few people being able to visit Human Technopole in person during this process, but many not being able to because of the different regulations in different countries. So I think both numerically and in terms of quality, I have to say I'm delighted by the standard and the quality of the people we've been able to hire. And I think they set a foundation for what can become a really successful research institution. The equipment, these are pictures of existing equipment at Technopole. The equipment is really state of the art. And the intention, as I said, is to build up this equipment, and some of these facilities will eventually become what are called national platforms or piattaforme nazionali, and will be open to the community. We have started to produce, so these are just a few of the publications that have Human Technopole affiliation associated with them. As time goes on, obviously, more and more of the work that leads to these publications will be done inside the Human Technopole buildings, and more of the scientists will be at Human Technopole full time. I should say, just again, in way of introducing Alessandro, that he was the brave scientist who was the first scientist to come on site and to start working on site when there was nothing basically for him to do in terms of scientific production, but there were a million things to do in terms of helping to set up the Human Technopole. And he was one of the people who contributed most to this very early stage of the development of Human Technopole from the research perspective. So I mentioned that we will be engaged in training. There will be obviously internal training for PhD students, for internships, for postdoctoral fellows. And because our intention is to recruit principal investigators who have just finished their first postdoc, at least most of the principal investigators will be of this category. They also require mentoring and also training in how to lead a laboratory, how to work with a group of people successfully. And then there will be external training events, and I'll mention that we already have carried out some external training events, conferences, courses, workshops, symposia, etc., to the benefit of external scientists. So the first courses that we've organized have, of course, all been where actually until the technology transfer course were virtual, because it was the COVID period. It was impossible to bring people onto the site to do the training, but they were extremely successful. They were very oversubscribed, and we intend to continue to work on these areas and additional areas as we grow. And as I said, we're enthusiastically engaged also in working with industry. And one of the courses that we have organized thus far, together with partners from Pavia and from Netval, was a course on innovation for researchers. And again, this is something we will continue to do. In terms of technology transfer, there are a large number of different ways in which we will work with industry. I won't go into this, but we were given, on the basis of the programs we had developed, we were given some financing to create an organization to help promote technology transfer in Lombardy. This is called the Center for Innovation and Technology Transfer. It's mainly funded for the use of Lombardy, but in fact, we wish to create this as a national facility, and this is now being set up. So a final initiative I'll mention, because it may be interesting for some of the people in your institutions, is that we've set up something called an early career fellowship program. This is a program to which people can apply who wish to carry out their first work as a principal investigator in an institution in Italy, not in Human Technopole, but in other institutions in Italy. We had the first selection last year. We had many good applicants, and we chose the first 5 fellows whose pictures you see here, 3 females and 2 males. This group are all Italian nationals who are coming back from other countries to start their own laboratories in Italy, and we're providing them with support to make this process possible and to make it a lot easier. We have opened a new call for the second group of early career fellows. This is not only open to Italians who want to come back to Italy, it's open to nationals of any country who wish to work in Italy. And so I would encourage you to encourage young people you are aware of in this stage to apply to the program if they want to come to Italy. And finally, I'll mention a few of the big scale projects that we're carrying on in relation to health issues. One is actually a cohort study rather than being focused on any specific health issue, but the Molisani cohort, for those of you who don't know it, is a very, very well organized and long-standing cohort in the Molise area of Italy, which has a huge collection of very useful data, but no genetic or genomic information. And so we will contribute the genetic and genomic information to this to make it an even more useful cohort. Then there's a smaller cohort based on patients suffering from autism spectrum disorders. And we have a couple of COVID-related research projects with various universities. And then this is simply to say where we are in setting up the national facilities. This is a process which we're doing together with 2 of our funding ministries, the Ministry for Health and the Ministry for Universities and Research. And we're at the level of the second level consultation, which is a general consultation of the scientific community about which facilities they would find most useful. So I will close there, and thank you very much for your attention.
Thank you very much, Dr. Matash. This has been a wonderful overview of what Human Technopole is. And I must say, from your exposition, there are, I'm sure from our audience, a lot of questions that we would like to ask you and to tell you about our everyday doing in research here in Mexico. But this introduction is wonderful, and I remind you that this presentation will be left recorded at the site of the Instituto de Biotecnología, so that you can tell our fellow scientists in Mexico and in Panama that they should really listen to this wonderful presentation. Before going on, Dr. Matash, I just wanted to tell you about one of the comments that was put on YouTube, because it is relevant to what you just said, and it is from Santiago March, from the Mexican Health Foundation, FunSalud, and he states that they will be more than happy to explore projects with Technopole and other Mexican institutions and research centers. But basically, he says that they are very interested in projects related to rare disease, data-driven genomics, proteomics, and pharmacogenomics. I just leave it like this so that you take this information in mind, before leaving. And also, before I say, I turn into our next guest, I would like to absolutely invite you to come to Mexico and visit us. It will be a great honor to receive you here, to show you our facilities, and to have a much closer interaction than what we have had until now. I hope you would have the time.
I would be delighted to do so, as Emilia has too much experience of this. Is the finding calendar that fit together is always the problem, and that's why we're doing this meeting virtually. But I would be delighted to visit Mexico at some point in the future, and I'm sure Alessandro feels the same way, and Maria Grazia, who will also be talking later in the meeting. So thank you very much. And I should say, if there are any problems with the presentation, we're happy to send you also to any of you the presentation directly if you just get in touch. So thank you.
Thank you. Thank you very much. Yes, and now we turn to our next speaker whose work was already introduced by Dr. Matash, but this is Dr. Alessandro Vanini. It is great to hear that you were the first one to jump into this wonderful project, Dr. Vanini. He is a biochemist and he heads the Center for Structural Biology after almost 8 years as a principal investigator and deputy head of division at the Institute of Cancer Research in London. His laboratory focuses on structural and functional analysis of large macromolecular complexes assembling around RNA for 3 loci and that play a role in gene expression and structural organization of the eukaryotic genome. These mechanisms are often deregulated in human diseases such as cancer and congenital neurodegenerative diseases. So without further ado, and with the introduction already had from Dr. Matash, please, it is my pleasure to introduce and give the floor to Dr. Alessandro Vanini.
Thank you, Marta. I hope you can all hear me well. I'll try to share my screen. I hope you can see it properly.
Yes, we can see the presentation.
Okay, you should also see maybe a laser pointer that moves.
We do.
Yes.
Congratulations.
Excellent.
Fixing that.
I've earned the next level of Zoom proficiency with this. No, thank you very much, Marta, for the very kind introduction. Of course, would have been lovely to be there in presence. I have fond memories of Mexico. I've never been to Panama, but it's probably a good occasion for the future. So what I will try to do today is just to give an overview of the center. I think, and I hope that I left the presentation broad enough for everyone to follow. I'll give a little bit more juicy details that Ian didn't disclose yet. So we will go a little bit into details of what we are trying to do and what we have set it up, but hopefully broad enough for all of you. I spent my entire postgraduate career abroad. So I spent 8 years, 7.5 years in Germany and about 7.5 years in the UK, where I basically started my own lab and became deputy head of a division at the Institute of Cancer Research. My lab in London is now is actually closing at the end of the month, so it has been phased out because finally we managed to build up all the labs here at Human Technopole. So my activity is going to be 100% here. And what I wanted to say is really that for me, coming back to Italy was not really an objective per se in my research career. I was quite happily where I was, but the magnitude and the vision behind the idea of Human Technopole really convinced me very quickly that this was once in a lifetime opportunity that I had really to catch. And of course, doing it back in your home country where you can give back I studied here, my university was here. It's also, of course, an element of, pride and additional value. But it is the science and the vision behind it that drove me back to Italy. And so what we want to do, really, the overall aims for our center is to use structural biology to discover and illuminate the mechanisms that really define human biology. We want to understand human biology better at the mechanistic level. And to do this, we want to do it by looking at mechanisms that, of course, are deregulated in particular diseases. We, as a center, we are not focusing on specific diseases, but on very central molecular mechanisms that underlies human biology. And the other really big aim, I think, is that is really much needed, I think, especially nationally in Italy, is to really provide an infrastructure for structural biology. So to enable the larger community of structural biologists, but also of molecular biologists, which is very solid here in Italy, to have access to the latest technologies, but also to the know-how. And so networking and training will be a fundamental aspects of our aims. And so I believe that infrastructures are fundamental, but you can have the best microscope in the world, but if you don't know how to use it properly, if you don't know how to prepare samples to arrive to the point where you can really benefit from those infrastructure, you won't gain much. You will actually just spend a lot of money and without much outcome. And so what you can see here in this slide, which I took from our colleagues from the Neurogenomics Center as an example, brain and central nervous system cells, is that if you want to really understand life, cellular life, you want to image it. I'm very much a believer of of believing in what you really see. And so you can go from very detailed atomic structure of proteins up to higher complexity into a full developed brain, and passing through intermediates in terms of complexity and also dimension of the spatial space that we are exploring. But and we are relatively good at doing all these steps in isolation, right? What we really are missing is how we correlate these to each other. How do we put an atomic an atomic resolution structure of a protein or a complex within its cellular context, and then even more so into a more complex system. So there is this spatial scale that we really want to try to unify. And the same is true for the temporal scale. We usually are quite good at take individual snapshot of a cell or a protein or even an organ at a specific time and in a specific circumstances. But what is really needed to understand life or cellular life at scale is really to overcome these two major issues, which is bridging the gap between reconstituted in vitro very detailed structures into more and put them into context, correlate them to their space and to their position into cells, tissues, eventually organs and tissues from patients. And the same is true also for the temporal control. We look at this, as I said, at specific snapshot. What we would like to have, it's something which is more dynamic and represent what is happening at an atomic level, but also on single cells and in tissues during the different time scales. And so those are the major challenges that we want to break. And this we want to do this by using, let's say, three pillars in our center, which is, of course, the first one is to do excellent science. So science and the level of science we want to do is as the priority. We are, as I said, not focused to one specific disease, but anything that will enhance our understanding of human biology it's welcome in our center. And so this means that we can also use model organisms to help understanding human biology. There is this concept of translating what we see in vitro, so as per recombinant reconstituted complex, also in situ. I'll touch upon what in situ means in these days in structural biology. We will have to be disease oriented. So we want, in a way, to understand biology, but also take the opportunity to be in this fantastic environment. And also, when I talk this fantastic environment, I'm talking about also the community at large in the Milan area, in Lombardy, to really then eventually translate or push some of our basic discovery into something more translational. And for doing that, of course, because of the broad scope and actually the challenges that we have, you can only do it by using really team science. So we have to collaborate with groups within Human Technopole, outside Human Technopole, to really answer those biological questions at large. Technology is, of course, very important. I'll show you a little bit what we are building up. Of course, you can have brilliant ideas and you can have brilliant scientists, but if you don't have the right infrastructures and the right toys to play with, if you want, you are then limited, and that's obviously a problem. And I think also what Ian mentioned, the fact to be open to external access. Yes, this is of course very difficult to implement properly, because balancing between in-house research and external research is a challenging task. However, the opportunities to actually be much more open to understand what is happening in a larger community, and to be always reactive to what are the needs in terms of technology and in terms of the biology that needs answer. And again, the third pillar is of course training and support. We wanna really be good trainers for our own researchers and people that maybe will develop a career as a technologist, as a support unit manager, and really being in charge of these facilities. And we want to do it again for our internal employees and researchers, but also for the externals. And in that respect, it's also really important for us to develop specific academic and industrial partnership. There is a value in talking to neighboring universities, of course, but also to neighboring companies, for example. And so we were very lucky, or maybe I want to say we were very good at hiring the first group of group leaders. So we are now 5, including my own lab, and we cover quite a broad space of biology. As I said, my lab is interested in, let's say, to put it in a very general way, let's say nucleic acid transactions that happen in the nucleus and sometimes in the cytoplasm as well, with the idea to look at non-coding gene transcription and also how this contributes to genome folding and regulation. Francesca, which is an early winner of one of the ERC starting grant, which is one of the most prestigious grants at the European level, is really focusing her structural biology approach to dissect the molecular mechanism of thyroid metabolism. So the thyroid is, of course, an organ which is very important for human health. There are number of dysfunctions that, of course, affects patients, cancer patients and other patients with thyroid dysfunctions. Philip Erdmann is a group leader that comes from the Max Planck in Martin Street in Germany, and he is really one of the first pioneers of a technique which is called fib-seb milling, which means basically it's a technique by which you can use a special microscope with an ion beam that allow you to create basically a window inside the cell and start to image, let's say, these macromolecular complexes in situ, as I said before. Gaia, which is my deputy, if you want, or associate head of the center. Really, her lab focuses on the structure of function characterization of cilia, and cilia are very important as in almost any cells, and there are a lot of ciliopathies that underlines a lot of human disease. So Gaia is obviously a world leader in this, and use a structural biology approach to understand that. And last but not least, Anna Casagnal, which is focusing her research on RNA editing machinery. So we now know that modifications of RNAs are really important for stability of RNA and impact on several cellular processes, and ultimately have been discovered to be important for many in many diseases, such cancer and neurodegenerative diseases. And so, well, the type of work we do, for example, is as I said, we are always very, let's say, focused on what is then the relevance of this molecular mechanism into disease. One example is one recent paper from Gaia Pigino's lab, where they figure out that the specific modifications of tubulin and a component of the cilia, or well, in this case, the flagellum of a sperm cell, was actually really relevant for the correct function of the flagellum of the sperm, showing that patients that have mutations in enzymes that do these modifications have actually sperm which does not move correctly, giving rise to infertility. So again, very detailed snapshot at the molecular level of what causes at the end a disease or a dysfunction. In our own lab, we have revealed the three-dimensional structure of the human RNA polymerase III machinery, which is the transcription machinery that transcribes all tRNAs, for example. And what we could do there is to map the specific location of genetic inheritable mutations that are either causing very severe neurodevelopmental disorders. So those patients do not develop a normal central nervous system, and they are very, very sick, unfortunately. And other mutations that are then involved in actually hypersensitivity to certain viruses. So again, as I said, we really try to push the molecular mechanism that we find in vitro to have a relevance for disease. In doing so, we have developed collaborations locally, for example, to develop a cross-link mass spectrometry platform, as we are now approaching a stage at Human Technopole where we are currently limited with space. We thought to partner with the neighboring institution and to set it up together. We are developing new workflows also with companies, for example, with Lumix that produce this optical tweezers machine. We are now collaborating on trying to develop a new way to move samples from their machines to a cryo-EM microscope. And as I said, we are also developing our strategy to open the access of our EM facility to external users. At the moment, we are hosting, not on a regular basis, visitors, but whenever we receive a request from an academic, not a collaborator, but an academic colleague, we always try to accommodate their visit to us to learn new techniques or to be trained on certain aspects for which we have already quite a good knowledge. And so to do this, of course, as I said before, we need a lot of infrastructure. The major part is here. So one of the things that is quite important for us is the biomass production. So we produce recombinant complexes or endogenous complexes by accumulating large quantity of biomass. And so we can produce regularly. We have a support unit that will help us, our researchers, to, for example, produce high quantity of proteins in insect cells. And so there is a constant culture that goes on, and researchers do not have to worry about setting up the experiment, but they'll find always those cells available there. We have medium scale fermenters for large quantities of yeast or bacterial cells. So sometimes the complexes that we want to study are so low abundant that you really need to start with large volume of organisms to really study them. And this is a setup which is not trivial, but now it works brilliantly, and so we are not limited by the amount of cells we need. We can do this also for mammalian cells. So we have a system to do mammalian cells in suspension to up to 10 liter with a bioreactor that allow us to grow cells at very high optical density. There is a very sophisticated way to collect these cells using acoustic waves. So you have a perfusion system where your cell culture goes on perpetually, basically, as long as you can pay for the media. But that's how it works. And so we have, of course, biophysics. So all a set of fancy equipment to characterize protein complexes from, an army of systems for protein purification, and all sorts of toys for characterizing proteins at the single molecule level, but also to understand or quantify interactions for proteins. For example, here, we still have a platform for crystallization. X-ray crystallography was the predominant technique in structural biology for the last 50 years or so, but in the last 10 years, it's clear that EM, electron microscopy, is becoming really the state-of-the-art technique. But I think it's still important to have the possibility to set up crystallization trials, and we have regular access to the synchrotron through an Italian block allocation group, and so we could do that as well. And so we are also investing in optical tweezers. So this is a machine from Lumix in particular. So this is a relatively easy to use confocal microscope coupled with the quadruple optical tweezers. So you can manipulate everything, and you can trap in between optical tweezers, and you can quantify, but also look qualitatively at molecular events at the single molecule level. For my lab and other labs, especially involved either with microtubules or with DNA, this is relatively trivial to trap those things. So you can it's really a nice system where you can look at events happening and at the same time quantify it. So that's this qualitative and quantitative approach, which we find very helpful and integrates pretty well with cryo-electron microscopy, which, as Ian was suggesting, is really what we heavily invested on. And so this is what we currently have. So it's 5 microscopes. It's quite a broad range of techniques that we can use. I want to really mention Paolo, which, as you can see, had a lot of work to do. So this, you have to remember that setting up this facility all happened during pandemic, right? So during serious lockdowns, the first lockdown here in Italy was in 2020 was quite serious. So Paolo had really sort of really a difficult job to do it, but he did it, and we have a perfectly functional facilities. Those are quite expensive microscope, expensive also in terms of maintenance. And of course, you want to get the best out of it by constantly using them to have the right samples to put in them. And what you should be able to see here is actually a time lapse of what it takes to build one of those big ones. Paolo took part himself in the building, but the end result, beside these very fancy pictures of the facility itself, is really, this is a sample that we collected here at our facility. It's a standard sample, but you can really see that the resolution for the biochemist aficionados here, you can really see almost the atomic structure of side chains of a protein, right? So we are talking about 1.5 angstrom resolution. So you can really study these complexes in high details. And what you see here instead is the usage of this other microscope, which is not just a microscope, but I should be able to pause it or to run it again. But what this microscope does is really to ablate material from a frozen cells, for example, and create what you see here is called a lamella. So it's a very thin, you can also write fancy things at the nanoscale, but this is just an example. But what you create is a very thin lamella, which is somewhat a very thin section of a frozen cell, where you can then go on one of the fancy microscope that you've seen before and image your complex of interest in situ. So exactly as a portion of a cell, so as a slice of a cell. So what you learn there is not just the structure of the macromolecular complex itself, but this entire neighborhood. So how it's spatially related to other complexes that are around, and so forth. And I think in the future, this will become more automated, more common, and will probably complement pretty well with what is called spatial mass spectrometry, where there are, of course, efforts to go and carry out mass spectrometry measurements of very, very specific and confined spaces within a cell. Okay, so just to recapitulate, that is my just final slide. I hope that I gave you an idea, a general idea of what we want to achieve, and you know, it's also in a way to remark the fact that to again to have a bold vision and to push science forward, we need technology. Technology is expensive. We need human resources, in the sense that we need good people. Again, you can have one of these fanciest microscopes, but if you don't have a good driver, that microscopes will never perform as it should. And equally important is to have the right samples and the right biological questions to answer. So we have to work at large with the community to really, in a way, enhance the understanding of structural biology. What are the answers that can be, what are the questions that can be answered, and how to arrive to the point that we can make the best use of these microscopes. And with that, I conclude, and I thank you very much for your attention.
Well, what a fantastic kickoff of the Italian Research Day 2022 you have given us. Thank you very much, Ian and Alessandro, for this wonderful presentation of Human Technopole. And with this, we finish the first part of our program. And I believe everybody is excited about everything that you have presented today. I would like to give the floor now to Maria Grazia Magro to continue to the second part of the program of today. And I thank you very much. I was honored to moderate this first part. Thank you.
Hello, Emilia, can you hear me? Or hello, Marta, sorry, can you hear me?
Yes, yes,
yes.
We can hear you.
Okay, sorry, I'm having a bit of a, I hope to be able to have my camera working in just a moment. Apologies. But in the meantime, I will just start by saying that I'm really thrilled and delighted to have been invited to this event. So I want to thank the organizers, first of all, for having this event, which is a great opportunity to make Human Technopole more known, you know, throughout the world, and in this case in Mexico and Panama in particular. And as in my role as head of strategy and scientific affairs at Human Technopole, one of the areas that I take care of is that of collaboration with scientific institutes in Italy, both in Italy and abroad. So this is really the type of events that I love to participate in and to promote, because more often than not, really very nice links and opportunities for collaboration come from them. Okay, I will just start without my camera because somehow it was working fine before, but now it's not authorizing Zoom to use the camera. So I will just start by introducing the first speaker. And the first speaker is Miriam Solis. So, or Mayrim Solis. She's a senior health researcher from the Gorgas Memorial Institute for Health Studies in Panama. She received her PhD in biotechnology from the National Cheng Kung University in Taiwan in 2016. She stayed on in Taiwan for 9 years as a stem cell researcher and returned to Panama to join the Gorgas Memorial Institute. She's currently a PI in the stem cell research group at the Gorgas Institute, and she leads 5 different projects having to do with diabetes and COVID-19. She was awarded among her recognitions, let's say, the L'Oréal National Prize for Women in Science, the Enid Cook Scientific Medal, and several other recognitions, including the Keys to the City of Panama. She's member of several national boards and of the Panamanian Association for the Advance of Science. So with this, I would like to leave the word to Mayrim, and we look very much forward to listening to her talk. Thank you very much.
Thank you very much for this kind introduction. Thanks for the organizers for giving us this great opportunity as a government research institute to have these doors open for collaborations for our national researchers and try to make good science and interesting science go on in our country. I really appreciate all the organizers, Dr. Emilia for all the logistics and organization and contacting us, and Dr. Pascal for allowing our research group among all of the great research groups in our institute share the research we are doing recently in Panama. So with this, I will share screen. I'll share my presentation. I hope everybody can see it.
Yes, we can see it.
Okay, I'm trying to, okay. Okay, so in this occasion, we would like to share the recent project that we have developed after the pandemic was declared in our country regarding the effect of COVID-19 in a well-established platform of stem cell research in our institute. Gorgas Memorial Institute is a government public institute, the only research in the public government level in Panama that is in charge of doing all of the vigilance and all of detections of positive cases in Panama, and with this try to train local hospitals for the detection of COVID-19 and other emerging diseases. Beside that, we have the role of doing research. So we have several research departments in genomics, virology, parasitology, including the stem cell research that I have been able to lead and to direct inside of our stem cell research group. So as soon as the pandemic was declared on March 11, only 8 days after the declaration of the pandemic, we were already approved institutionally for starting the research that I want to share. Okay, they're telling me that I'm not projecting the whole screen.
It's on presentation mode.
It's like, in presentation mode?
On presenter mode, not presentation, if that makes sense. Like we can see your PowerPoint.
But do you see the slides in the side, or do you see the whole?
On the side, we see them on the side and on the middle, yeah.
Okay, sorry for the technical issue. I'm trying to. So, sure.
No.
Sorry.
We can see it, but we don't see the full screen. Okay.
Okay.
Okay,
Now we can see it.
perfect.
Thank you. I'm sorry. So only 8 days after the pandemic, we were, let me put this, okay. Only 8 days after the pandemic, we were able to have the institutional approval for starting a study in order to evaluate if COVID-19 was, or SARS-CoV-2 was transmitted vertically from the mother to the baby through placenta evaluations, and at the same time try to understand the effect of COVID-19 in the mesenchymal stem cells that reside in these placentas. So we started a very we got the approval from the IRB National in Panama of bioethics, and after that we started to during the lockdown of the country, our research group activated and keep activated in recollecting the placenta from these COVID-19 positive patients. And by April 30, the first case of SARS-CoV-2 positively detected in placentas was reported in Switzerland. So with this, by that time we already had several placentas already collected, and by August 25, already 34 cases of SARS-CoV-2 detected in placenta was detected worldwide. By January, we were able to report our first 2 cases of SARS-CoV-2 detected in the placenta of 2 women in our local hospitals, which was detected by July 2020. So with this, I tried to explain the timeline, because during this screening, we were on the time in the same track of trying to see what happens in our populations and with the lineages of SARS-CoV-2 that were collected, that were detected and sequenced in our country. So at this time, we already knew that pregnant women had the same risk of could be positive to COVID-19, and there was a very high rate of these pregnant women to be delivered by C-section and have preterm deliveries. By this time, there was fairly convincing evidence that SARS-CoV-2 was transmitted from the mother to the baby, and it was not a very usual event as occurs with, for example, SARS-CoV-1 or Zika virus. So even though this was fairly convincing, as I just say, we already were actively recollecting the placenta from these patients, and we after several placenta screenings, we could observe 2 cases with CT values by real-time PCR below 35 that indicated positive detection of SARS-CoV-2 of these placentas. We could see the amplification plot, the melting curves, and we can indeed see these 2 cases were detected by real-time PCR compared with all of the other placentas that were detected negative. And the Department of Virology, led by Dr. Sandra Lopez in collaboration with us, we started to isolate the virus, the SARS-CoV-2 that was present in these 2 placentas. And we indeed were able to isolate the SARS-CoV-2 in our BSL-3 laboratory in our institute, and we infected this isolated SARS-CoV-2 into viral cells, and we could indeed see the cytopathic effects. So after we isolated this virus, we ran real-time PCR again, and we can indeed see the positive detection of this SARS-CoV-2. So with this information, we were able to report for the first time in Panama the 2 cases of SARS-CoV-2 detected in the placenta of these 2 patients that were reported clinically as asymptomatic. They were able to be detected for COVID-19 positive because for institutional regulatory, it was mandatory to have a test of COVID-19 in order to be admitted to the hospital. So we were able to observe by pathological studies that SARS-CoV-2 was localized in endothelial cells. It was a very important piece of information in Panama and worldwide, because it is already known that the vertical transmission of SARS-CoV-2 from the mother to the baby usually occurs when SARS-CoV-2 localizes in trophoblast cells. So this is one of the possible indications of the reason why the babies from this mother were detected negative just after delivery. These SARS-CoV-2 viruses were sent to the Department of Genomics, led by Dr. Alexander, and we were able to detect that the sequences that presented these viral lineages. And we could observe that the lineages were A2B1, and both placentas, both viral lineages contain the same mutation of G11803T, which is our already reported mutation which role is in adapting SARS-CoV-2 to the environment. So with this, we had a chance to contribute to what was known by that time worldwide in the global worldwide repository of information, and report what was happening in our country regarding the detection on the vertical transmission of SARS-CoV-2. But the next question was, if there's a possibility of having SARS-CoV-2 invading the placenta, what happened to the population of mesenchymal stem cells that we have been working since 2017 in our institute? So we tried to put our information, all of our effort of research in what we have already established in 2017, in which we already had routinely our SOPs for the isolation of stem cells, mesenchymal stem cells from placenta. We have a quality control assays, very robust in our institute. We have a stem cell banking that serves us for preserving all of the stem cells that we will be isolating from these COVID-19 patients. And we have 5 different research projects focused either in diabetes or in COVID-19. So putting this information together, and what we were already observing in literature, in which placenta stem cells indeed play an important role in suppressing T cell proliferation inside the placenta, and that the first clinical studies of mesenchymal stem cells in COVID-19 were started to be reported in China by January 2020. Then we were able to start the recollection of placentas from COVID-19 patients during the local lockdown. So after this, this is a manuscript that we have undergoing for publication, and we were able to observe interesting results. Whenever we see COVID-19, that is the left picture, the top left picture, we could see that non-COVID, let me see if I can put a pointer. Okay, I don't know if you could see the pointer, but we could see that, no pointer can see. Puntero láser. Okay. So we were able to see in this picture, this is the normal morphology of stem cells from non-COVID patients we have, which is the normal size that we observe routinely. But whenever we isolated the stem cells from those patients in the same passage, P4, and we isolated them from these COVID-19 patients, we started to see just by the morphology, the size was much more expanded in their cytoplasm. We were also able, because of already theses that were undergoing in our research group, we could compare a very similar size of the stem cells from COVID-19 patients to the size of those stem cells that were cultured for 18 passages. So there was something interesting going on the stem cells that were isolated from the COVID-19 patients, and our quantitative results supports this information of a bigger cell size in the stem cells from COVID-19 patients compared to the non-COVID-19 patients. One of the important abilities of mesenchymal stem cells is to form colonies, and we started to see that stem cells from COVID-19 patients have less ability to form colony compared to non-COVID patients. Besides that, we started to analyze different mechanisms of proliferation, and we observed that COVID-19 patients had higher proliferation rate compared to non-COVID-19 patients. And beside that, the ability to differentiate to different lineages such as chondrogenesis and osteogenesis, we could see that as soon as we isolate the stem cells, there was a higher ability to differentiate, but this ability was turned off after just several passages. So that was some information that we were starting to recollect and we find to be very important to report and to publish. And then, but we still wanted to see furthermore, we started to infect in our BSL-3 facility SARS-CoV-2 into stem cells from these populations. And we could observe that if we infect stem cells to the COVID-19 patients, the changes are not as dramatic as we infect this virus to non-COVID-19 patients. We also did a MOI assay at different multiplicity of infection of SARS-CoV-2, and we observed that whenever we infect a different MOI SARS-CoV-2 into COVID-19 patients, we will see not very significant changes. We compared 3 different moms in this slide, and as you can see, in the 3 cases, not significant changes in the proliferation of these cells or the cytopathic effects. But whenever we infect at the same MOIs to COVID-19 patients, to non-COVID-19 patients, to non-COVID, we started to see a dramatic cytopathic effect. So we could see that indeed there's something going on in the stem cells from the COVID-19 patients. In regards to their ability to differentiate, we could see that the differentiation ability to chondrogenesis was reduced in most of the advanced passages, and even after infection, the ability to differentiate was turned off. So with this information, we were able to go on to understand the molecular mechanism. We wanted to understand what is happening in the signaling of the SARS-CoV-2 in our stem cells. So we ran immunofluorescence assays and several Western blots of the most common reported receptors of SARS-CoV-2, which is ACE2 and TMPRSS2, and we observed that compared to our normal CD marker, CD90, we will and compare as a positive control our ACE2 in viral cells, there was absence of ACE2 in our mesenchymal stem cells. By Western blot, indeed, we confirmed that either stem cells from non-COVID patients and from COVID patients, we will see no expression of ACE2. So now the next step is to understand what is the signaling mechanism occurring in all of the phenomena that I have just presented. So we started to develop techniques in CRISPR-Cas9 for knocking out common receptors that have been already reported in literature. It has been already reported that SARS-CoV-2 infection reveals an ACE2 independent inflammatory activation of endothelial cells, and this occurs to toll-like receptor 4. So now one of the researchers, member of our group, which is Cindy Fu, which will be having her master's degree in Italy in September in the University of Calabria, she's starting to understand what is the signaling mechanism occurring through toll-like receptor 4. And another research member of our group, Erika Guerrero, is right now training in Holland, Netherlands, in order to understand and to create the techniques of CRISPR-Cas9 for knockout and knock-in, and try to establish these experiments in Panama in Gorgas Institute. How we want to do this? Well, first we are trying to confirm the signaling that occurs through toll-like receptor 2 and 4 by Cindy, and then we are trying to apply the CRISPR-Cas9 vectors to our cells. So for instance, we have already designed the toll-like receptor 2, toll-like receptor 4 in our database for vector selection. We will be performing the annealing, the ligation, the digestion into the lentivirus that we will be using for for the infection, and we will be transforming this into the bacterial cells and confirming that indeed we have succeeded in the construction of our CRISPR system. So after we have this constructed, we will put it together with our Cas9 guide sequencing of RNA and transfect it with our lentivirus by electroporation to our PDMSE and try to confirm if indeed we have knockout by Western blot our toll-like receptor 2, toll-like receptor 4, and then we hope to see if there are any opposite effects of what we have been observing on the stem cells and COVID-19 SARS-CoV-2 effects, either naturally or in vitro. So with this research, we are trying to put some information, not only for Panama, but worldwide, in order to understand what is the effect of SARS-CoV-2 signaling on these placental cells and what impact will have in the pregnancy status of patients. And of course, looking forward into stem cells be a possible therapeutic strategy for COVID-19 patients in our further studies. So I would like to thank our research team because you see that only 8 days after the pandemic, we put together this research team with virologists, biotechnologists, obstetrician for the placenta recollection, pathologists, and the genomic center in order to try to collaborate worldwide to this pandemic that it has really tried to put some important information worldwide. So I would like to thank our financial institute, Gorgas Institute, and our stem cell research team that which you see it's very small, but we are trying to do great work that we hope we could share and collaborate with Technopole. So thank you for your attention.
Thank you very much, Myrim. And you should be able to see me now that I've solved the technical glitches I apologize for. This was a very interesting and very current talk, obviously. We would like to move on now to our next speaker. Unfortunately, we are a bit behind on schedule, so I really ask speakers to please stick to their 10-minute time limit. The next speaker is Dr. Tonatiuh Ramirez. So Dr. Ramirez holds a PhD in chemical engineering from the Drexel University in Philadelphia, in the USA, with a specialty in biochemical engineering. He, until recently, he has been the Director General of the Instituto de Biotecnología, so the IBT UNAM, and he has, as such, overseen all of the academic and administrative activities of the Institute. In his role as Director General, he also very successfully fostered the translation of several technologies into the industrial sector. So he obviously has a very keen interest in technology transfer, and this has also characterized his own research, which focuses on the cellular and molecular response to environmental gradients in industrial reactors. So his work has laid the basis for the design of several new engineered bacterial strains and cell lines, and he's achieved really important results in the production and characterization of recombinant therapeutic proteins for human and for both human and veterinary use. So the floor is yours, Tonatiuh. Thank you very much.
Thank you very much, Maria, for your kind presentation. And I also want to thank Emilia for her kind invitation to this conference. You should be now seeing my screen. Is that correct? That's a blue screen?
Yes.
Yes.
Yeah.
Okay, so let me start to saying good afternoon to everybody. And as you can see here, there's a nice picture of our biotechnology, the Biotechnology Institute of the National Autonomous University in Mexico. And today in this short 10-minute presentation, just as this picture shows you, I will try to give you an very extremely brief bird's eye view of the projects that are related to health in our institute. Okay. Yes. So our institute is one of the largest of our university with a community of more than 1,000 people. Of them, we're about 140 researchers working in these 5 departments: plant molecular biology, cellular engineering and biocatalysis, molecular microbiology, developmental genetics and molecular physiology, and molecular medicine and bioprocesses. In red, here I show the 2 departments where most, not all, but most of the health-related research is done. It would be impossible for me in a few minutes to give a good summary of what is being done by all of our researchers. But at least what I have been showing, what I'm showing you here in this list is some of the most important areas that are being addressed today. You can see that the biological models expand from mammalian and human cells to insects, rodents, fish, spermatozoids, the nervous system, virus, phages, and bacteria, among others. And the interests span many different areas, including developmental biology, genetics, neurobiology, nervous system, pathogen-host interactions, virulence, genomics, toxinology, immunology, neuroinflammation, and many others. Most of the basic science which we work on rarely ends up in some tangible application. However, here in red, I have highlighted some exceptions where some of our colleagues have been involved in all of the steps of development that finally reach the patients. These are scorpion, snake, and spider antivenoms, and recombinant biopharmaceuticals and vaccines. And to support all research at IVT and elsewhere, we have 11 core centralized facilities. In particular, we have 3 national laboratories for advanced microscopy, analysis of biopharmaceuticals, and technological support to genomic sciences. We have also a beautiful animal facility, as well as unit dedicated to proteomics, massive sequences, and bioinformatics, pilot plant and scale-up, DNA synthesis and sequencing, cytometry, and cell sorting, and plant tissue culture. So you can see that the pillars of our institute are very similar to those of the human Technopole. That is, we have a very strong basic science foundation of excellence, a search for translational science, central focus on teaching and human resource training, and exploitation of common core facilities. So now coming back to the cases where our researchers have been able to reach the patients. One of the first cases that our institute was involved was in the approval of a scorpion antivenom. This was the first Mexican pharmaceutical product approved by the FDA in the United States. And what you can see here, it's an actual picture of the Times Square in New York, the day of the approval. This work was done by Alejandro Alagon and the companies Silanes and Bioclon. In addition, with Inosan, another company, today, more than 800 patients are treated daily worldwide with some antivenoms that Alagon helped develop. For instance, thanks to these phagotherapics, mortality in Mexico due to scorpion stings has decreased by 1 order of magnitude. The other example comes from my group, which was a key participant in the development of ProBiomed, the first pharmaceutical company in Mexico to produce therapeutics and prophylactic recombinant proteins with the highest national integration from gene to product, and contributing hundreds of millions of doses of biopharmaceuticals at accessible prices in Mexico and many other countries. For instance, here I'm showing you how erythropoietin prices substantially dropped with the introduction of the biosimilar version of erythropoietin of ProBiomed. Also, the group that I conform with Dr. Laura Palomares has been key in the inclusion of Liomont in the biotechnology arena. Liomont is one of the largest Mexican pharmaceutical companies who launched its first biotech product in 2015 with our support and technology. It has now 4 biosimilar monoclonal antibodies in the pipeline and a state-of-the-art biotech plant, which we design, start up, and operate for engineering, technology transfer, and validation runs. So as you can see here, our institute has a long tradition of engaging with the private and governmental sectors, as you can infer from the many logos of companies which we have had some type of relation. Most of them, as you can see, are companies related to the health area. However, what is relevant from the companies that are in this first line is that our relation has ended up in specific products that have benefited the patients. With many of the other companies, our relation has been limited to specific services or limited research that never reached the patients due to some structural limitations that I will be addressing in the following slides. Such structural limitations prevent the closure of the virtuous cycle of science, technology, and innovation. And as you know, such virus cycle, which I indicate here with this picture, is based on converting a lot of time, a lot of science and money, and many, etc., to some little knowledge. This is usually and primarily done by academia in the initial steps. And then, as I mentioned, you need lots of this to generate a little of this. Then through innovation, primarily in industry, we can turn back knowledge into the well-being of the population. The problem in Mexico and in many other countries is that this cycle is incomplete, as key links are missing, specifically those that have to do with enabling human capacities and infrastructure that bring together academia, government, and industrial partners. In the past few years, our research group, in addition to the traditional academic work, we have also been working in contributing to solve some of the problems of these missing links. For instance, we have established a peripheral unit of our institute in the city of Pachuca in the state of Hidalgo, where a pilot plant and an analytical laboratory will be established. And with this, we will be contributing at least to 2 necessary but missing links in Mexico to close the virtuous cycle of knowledge innovation. Here, you can see the building that's already in place in the knowledge district, which altogether we will have a surface area of about 1,700 square meters. This represents a strategic alliance between the university and the state of Hidalgo and has been initially supported by the company Sartorius. As you can see here, the day of the installation of the board of directors, our rector was present along with our director and the government of the state of Hidalgo. The infrastructure will support the development of vaccines and biotechnological medicines, facilitating their transit from basic research to their clinical evaluation, and what is more important, improving the chances to reach the patients. The unit will offer technological services to national and international pharmaceutical industries, producing drug substance and finished drug product in a multi-purpose pilot plant operating under the highest international standards of good manufacturing practices. We anticipate that lots of at least 4,000 prefilled syringes or lyophilized vials will be produced, and the product manufacturer will be employed in clinical trials, among other uses. In addition, the pilot plant will be supported by our national laboratory dedicated to the analysis of biotechnological medicines and biopharmaceuticals, or as we know it, LAM. This is an overlay of the pilot plant, and I know it's very small for you to see the details, but I just want to show you some of the aspects of the design of our GMP pilot plant, which will count with the production of service areas as well as the production itself. It will contain an area for bacterial culture at the scale of 15-liter fermenters, messenger RNA synthesis, and culture of animal cells at the scale of 200 liters. It will also have extraction and purification suites and fill and finish areas of the biotech products. In this way, the pilot plant will have all typical unit operations of a bioprocess, from upstream to downstream, to generate the active pharmaceutical ingredient up to the finished product of the pharmaceuticals and vaccines ready for clinical use. The project considers a self-sustained operation with international standards available for both national and international players, and includes as a central aspect the training of highly qualified human resources, as well as to serve as a showcase for teaching society in general about the importance of science and technology. And in the lower level of this building, we will have the analytical laboratories, which will perform all relevant and detailed chemical, physical, chemical, and biochemical studies to determine the quality, potency, and safety of the products generated in the pilot plant. The second missing link, and with this I'll have 2 more slides to go, in which we have been contributing, is to develop enabling capacities, particularly in the analytical area. In the past 2 years, we have focused all our efforts in the fight against COVID-19. Specifically, we established a strategic alliance between LAMB, our national laboratory, which I referred in the previous slides, which contributes all its capacities for analytical characterization of biotechnological medicines and vaccines, with 2 other players, UNIPREC, which is the unit of preclinical assays of the School of Chemistry of our university, and which has a specialized animal facility and thorough experience in preclinical studies. And UNIDESMIC, which is the unit of the microbiological challenges of the School of Veterinary Medicine, also from our university, and contributes its biosafety laboratory level 3 and the experiences in its operation. We are very happy that the Coalition for Epidemic Preparedness Innovations, or EPI, as you know, which is located in Norway, has recently selected our laboratory as the 9th laboratory in the world to join their centralized laboratory network. This is an historic milestone for Mexico, since, as you can see here in this map, we are now the first laboratory in Latin America, which will join the first of 2 in North America, 2 in the United Kingdom, and 2 in Asia. We are also very proud to share with you that our director, Dr. Laura Palomares, was selected this year by CEPI to join their scientific advisory committee, and together with the Brazilian scientists, are the only Latin America representatives. The fundamental purpose of this worldwide network of laboratories is to evaluate vaccines candidates against diseases with epidemic potential, such as those listed here. Obviously, during the past 2 years, COVID-19 has occupied CEPI's main attention, and the central function of the group will be to offer in Mexico these 4 assays: ELISA for detection of antibodies against RB2, S and R proteins, neutralization of pseudotype virus, neutralization of wild-type virus, and cellular immune response by an ELISpot. These methods were developed by Nexalis and Public Health England and have been transferred to all the laboratories of the network following strict procedures to guarantee their reliability and reproducibility of results throughout the world. Presently, we are in the process of finishing the technological transfer, and within a few months, we should be ready to start providing the analytical services. With this, I want to close and look forward for the next section to discuss any possible scientific and technological collaborations. Thank you very much.
I was mute. Thank you very much, Donahue, for your talk. I will now introduce the next speaker. This is Ricardo Leonard Cruz. So Dr. Leonard he holds a PhD in biological sciences from the University of Havana. He actually spent most of his early career in Cuba. He actually spent 20 years at the Center for Genetic Engineering and Biotechnology, where he actually developed his career until the level of group leader. He then joined the Institute of Scientific Research and High Technology Services, the INDICASAT AIP, in Panama in 2008, where he's been working as a senior scientist. His group is particularly focused on the characterization of genomes of important parasites and the use of genomic information to discover new potential drug targets and vaccines. He's also been leading the virology laboratory, where the research instead is aimed at developing new diagnostic means and potential antivirals against important viruses, including dengue, Zika, and more recently, and unsurprisingly, SARS-CoV-2. So thank you very much, Dr. Leonard, and please, you also have 10 minutes for your talk.
Thank you very much, Maria, and I would like to thank Emilia and the Italian embassies. Can you hear me?
Yes, we can hear you fine. Thank you.
I'm going to see if I can share some slides. I wonder if you can see the slides.
Yes, we see them.
Well, thank you very much for your beautiful session. I want to thank you for this very interesting session, and I'm looking forward to establish collaboration between our institutes. This is INDICASAT. INDICASAT, I know it's a very complex, it's very difficult to pronounce acronym, but it means Institute of Scientific Research and High Technology Services. And we are located in the City of Knowledge. I don't know if any of you have been in City of Knowledge in Panama. Is this beautiful campus over here? It's just by one of the locks of the Panama Canal, and our building is right here, just in front of the Senate, which is here. And in the context of possible collaborations and next discussions, I wanted to present you some what we are, who we are, where we are, and what we are doing in research in Panama. We are a public interest association, AIP. This is why in our name you will see INDICASAT AIP. These public interest associations are a hybrid kind of legal figure in which we are not exactly a public institution, but we are not either a private institution. And by law, we are allowed to use sources from the government, from finance, and we are allowed also to manage private funding, something which is completely forbidden for strictly public institutions, even doing research. And by law, we have to be AIP, any AIP have to be under a public institution. In our case, it's the National Secretary of Science, Technology, and Innovation. We have about 52 scientists and technicians. Support staff is about 23. We have a lot of students, and we receive students from different levels of education. And for us, it's postgraduate students are the most important students. We develop some PhD programs in collaboration with international and national universities. We are a non-profit organization, and we devote our activities into three main branches: scientific research, capacity building, because we are engaged in some of our these PhD programs, and we also offer some technology-based services which are not available in the country. For example, we had to, during this pandemic, we had to reorient many of our researchers, and we had to start contributing to the national response activities to COVID. And in that sense, we participate in several projects together with other institutions in the country. And some of those projects I'm going to talk a little bit later in the presentation. We are organized, our scientists. We have at this moment a little bit more than 33 scientists. We are organized in several research centers. We have, for example, the Biodiversity and Drug Discovery Center. We have the Cellular and Molecular Biology of Disease Center. We have the Center for Neuroscience. We have a Center for Clinical Research and Translational Medicine, where they perform mainly clinical trials. We have an Innovation and Technology Transfer Center. This is a new center where we are trying to manage intellectual property and technology transfer activities. We also have an Academic Affair and Collaboration Centers, which is devoted to managing all the programs we keep with several universities. As I mentioned, we have PhD programs, collaborations for many students. We used to have it with some Indian university, and in the last 5 years, we have graduated more than 20 PhDs in biotechnologies through that program. And right now we are working with 2 programs, and one program with the University of Panama. It's an MD PhD, it's a PhD program for physicians. And we also have a program with the Technological University of Panama in biotechnology. In these centers, we have been doing several lines of research. We mainly work in biomedicine, in biotechnology, and biodiversity. And in biomedicine, we have been, for example, this is just a small sample of some of our projects. We have vaccine research and trials. In this case, we have been working on a project for already several years, trying to find vaccines against an important parasite in Panama, which is leishmania. We have been working in natural products and drug discovery coming from natural sources. We have been working in neurodegenerative and aging-related chronic diseases. We have been working in antivirals for detection and discovery of antivirals for dengue and SARS-CoV-2. And also we have researchers working in tuberculosis, malaria, leishmania, trypanosoma, and toxoplasmosis. In biotechnology, we have been working in biology and control of tropical parasites like Chagas, trypanosoma, malaria, leishmania, toxoplasmosis is a new addition to some of our researchers. And also we have a line of working with biomaterials, in particular, trying to see the effect of nanostructuring in compatibility with human tissues. This line of research. In biodiversity, we have researchers working in biology and ecology of social insects, in particular bees and ants. We have a group working in bioprospection, and we are going to talk a little bit more about that. We have 2 researchers working in systematics, ecology, and biotechnology of fungi, which is a rich source of new compounds. And we also have 2 researchers working in molecular ecology of vectors of arboviruses, in particular mosquitoes. In addition, we have transversal areas which are dealing with all the researches, such as, for example, genomics and bioinformatics. In the terms of capacity building and training, as I mentioned, we pay particular attention to our doctoral programs, and we have also programs with different levels of education. So we receive students here even from the beginning of their secondary levels. We also support a lot of seminars, conferences, and workshops, both nationals and internationals. Just to show you some examples of the research being done in our institute, this is some of the research being done by the natural product chemistry and biochemistry, and they have been searching the whole biodiversity in Panama, which is one of the hotspots for biodiversity. Looking for activities for a different number of tropical pathogens, for example, against leishmania, against malaria, against dengue, against toxoplasma, against other viruses. And they have been able to perform the whole cycle of the research from the screening of natural sources all the way through bioactivity guided isolation and purification and structural elucidation using very advanced tools such as NMR and mass spectrometry. We have in the Institute 2 NMR spectrometers, which are being used for structural elucidation. We have several GCMS spectrometers which are used for chemical identification and detection for some of these projects. In clinical trials, we have a unit not exactly located in our building, but in a different building closer to the hospitals in the near downtown. And at the moment, we are performing about 19 different clinical trials. I'm going to go quickly. I'm going to show you that we have an animal facility right now where we host both mice and rats, and we are able to provide SPF animals for the research both in our institute and in other institutes of Panama. In genomics and bioinformatics, we have recently acquired a high-performance parallel computer cluster, and also a genomic facility, which is going to be functional very soon. And with this, I would like to close, thanking the organizer and looking forward to having some collaboration between our institute. Thank you very much.
Thank you very much, Ricardo. Thanks a lot. Very interesting institutes, as some of the others that we've heard about. We really need to take that trip to Mexico and Panama soon, I believe. We're now about to, heading towards the end of this session with the last speaker, who is Dr. Selene Fernandez Valverde. So Dr. Fernandez is a research group leader at the Advanced Genomic Unit in the Cinvestav Center in Guanajuato, Mexico. So she completed her doctorate at the University of Queensland, studied new classes of small RNAs, and then stayed on at the same institute for a postdoc, or at the same university for a postdoc, focused on the use of comparative genomics to identify regulatory changes that facilitated the evolution of organisms and also morphological diversity in the animal kingdom. She has obtained many important awards during her career, some particularly impressive. She was awarded the National Scholarship L'Oréal UNESCO or AMC CONACYT for Women in Science in 2016, the International Rising Talents Fellowship, again, of L'Oréal UNESCO in 2018. And in 2018, she was also listed by Forbes Mexico magazine as one of the 100 most powerful women in Mexico. So she currently heads a group, or her lab, which focuses on the study and the evolution of long non-coding RNAs in various animals and plants, studying their evolution, particularly predicting their functions using bioinformatic tools. And that's what she will tell us about in her talk. So please, Selena.
Thank you for the kind introduction. So I'm going to share my screen now. 1 second, and that should work. And you should be seeing me now. So I'm going to try to stick to time as much as possible. So as mentioned, I am at the Advanced Genomics Unit Langevio, and I know most of the talks have focused on the capabilities there are. There are many, I will tell you very quickly. So we are based in Irapuato, in the center of Mexico. And we have numerous facilities. Most of the people at the Institute work in plants, in plant development. However, we have people working in biodiversity, in salamanders, axolotls, microbial diversity, and we use lots of genomics bioinformatic tools. And we have a very strong postgraduate. We have 2 postgraduate programs, one in plant biotechnology and one in integrative biology, with a strong bioinformatics component. And I will be telling you more about my work. So it's a very basic research question. However, it does have potential applications. And I'm sure people at the Human Technopole are aware of this, given they have Piero Carnici amongst their colleagues, who is one great authority on the subject. But just for a general public perspective, you must be aware that DNA gets transcribed into RNA, and then RNA becomes proteins. And one of the things that really interested me during my postdoc is the fact that proteins are very, very highly conserved. So they change very little through evolution. And for example, we shared like about 15,000 proteins with sponges, marine sponges, which diverged from other animals over 700 million years ago. However, we see lots of phenotypic diversity in very short evolutionary timescales. And we have seen that lots of that variation is actually not on the protein side of things, but more on the non-coding side of things, and particularly in RNA. So that's the topic of interest of my lab. And another thing that we have noticed with previous studies is that about 2% of human DNA codes for protein. However, 62% of it is transcribed. Thus, the question is, what is the rest of the RNA doing in its more simple form? And the molecules we study are called long non-coding RNAs, and these long non-coding RNAs are different from others you might be familiar with. So messenger RNAs, which are the ones that are translated into proteins, and ribosomal RNAs and tRNAs, which are also participating in the generation of proteins, protein synthesis. And there's other non-coding RNAs. So there is small non-coding RNAs, which there is a lot, and I won't talk about them today, but we're interested in particular in this group of long non-coding RNAs. And what are these? So they are RNA molecules longer than 200 nucleotides that have little or no protein coding potential, and they are similar to mRNAs, and they might or not have some of the chemical modifications that characterize these molecules. And one of the things I would say the thing that interests us most is the fact that they evolve very rapidly in a lineage-specific manner, and they're expressed in very few tissues or cells. And the function of the vast majority of them remains uncharacterized. However, the ones that have been characterized have very important functions. So for example, they participate in the establishment and maintenance of epigenetic modifications, so turning on and off many genes at the same time. And also in the regulation through antisense transcription, processing of the messenger RNA, generating small RNAs, which are also regulatory. And one of the most interesting functions that has been more studied in the last few years is protein scaffolding. So protein scaffolding is when an RNA is nucleating the union of many different proteins that don't have protein binding capabilities between themselves naturally, but they can bind through this RNA as a scaffold. And what I really want to point out is that between 60 and 80% of the disease-associated variants in the human genome are in non-coding regions. This is not only long non-coding RNAs, also enhancer and promoter elements. However, it's a very important fraction. And what I wanted to show you in this slide, this is a phylogenetic tree, and we can see, for example, here maize, Arabidopsis thaliana, which are plants. We see humans here, and all different types of human fish, a fish here, a fly here, etc. So what I wanted to notice in this graph is the fact that proteins, which are in pink circles, are a lot more retained, a lot more constant through this tree than long non-coding RNAs, which are in blue. In particular, when we compare different groups, they are very rapidly lost in comparison to proteins. So, however, we know that many of them are very important. So, for example, the long non-coding RNA exist participates in the silencing of one whole X chromosome in mammalian females, including ourselves in women. Each of our cells have one of the two copies of the X chromosome silenced. So then we have the same dosage as males, which who only have one single copy of the X chromosome. So we know that protein coding genes are very, very similar, and they have very strong selective pressure in evolution to not change, because it can be very detrimental and cause very serious diseases. However, long non-coding RNAs change very rapidly, and we don't understand how can they remain functional and be so important when their sequence just is very volatile. Thus, for example, in the case of exist, or in this case of this other non-coding RNA, megaman tuna, we see that they carry out the same functions in all these species. So this is human, mouse, and zebrafish. And however, many highly conserved regions are not essential, and only a few are very tiny portions of it are very essential. And in the case of 6, it's even more puzzling, because the highly conserved region is dispensable and the lowly conserved region is essential. So then what we generally use to tell that something is conserved, which is sequence conservation, does not work very well for long non-coding RNAs. So as I titled my talk, The Conundrum of Finding Non-Coding RNAs, to me, the conundrum is, how do we identify which long non-coding RNAs are functional? And for this, we are defining a biological function as the reason why an object or process occurs in a biological system, implying a benefit to its presence in a particular temporal or spatial context in that biological system. And molecules with biological functions are maintained through a process of natural selection. And we think we can help solve this mystery or try to identify functional non-coding RNAs using evolution. And I just want to quote this famous essay from Tadeusz Dobzhansky saying that nothing in evolution makes sense except in, nothing in biology, sorry, makes sense except in the light of evolution. And to shine this light of evolution of long non-coding RNAs, we are making a strong assumption that functional long non-coding RNAs must be conserved. However, the evolutionary signatures or dynamics might change depending on how they function. So if they function through structure or they function through rapidly co-evolving interactions, as is in the case of Exist. And so in my lab, we study non-coding RNAs from a number of different approaches, but today I'm going to be focusing mostly on evolutionary approaches. And so we have worked on finding different types of conservation that go beyond sequence conservation. So this is conservation of splice sites, conservation of small motifs, and also structural conservation. So this is to say that even though two non-coding RNAs might not look the same from their sequence, if they maintain the same structure, so they have the same pairings, then they might be functionally homologous. And this is work by one person in my lab, Jose Antonio Corona Gomez, and some of it is already published. And also we are studying this question from the sequence motif conservation section and the conservation by asyntony. This is a non-coding RNA that emerges from the same site, but it's we cannot find a lot of similarity between different species, might still retain the same function. And our final aim is to develop computational methods to identify non-canonical long non-coding RNA conservation in different groups. We also have a project funded by the Royal Society in the UK, which I cannot tell you too many details about because of time, but we are surveying RNA expression, RNA-DNA interactions, and 3D nuclear DNA formation, aiming to identify long non-coding RNA-DNA interactions that contribute to the establishment and maintenance of chromatin architecture and gene expression. And another project I wanted to tell you about, given the interest, of course, in humans, in the Human Technopole, is another project we have in collaboration with the Mexican Biobank. And this is a project led by my colleague, Dr. Andres Moreno Estrada, he's literally my office colleague, like he works at Langevio with me. And Dr. Maria de Lourdes Garcia Garcia from the National Institutes for Public Health in Mexico, and Professor Adrian Hill from the University of Oxford, and many, many, many other people like the University of Stanford, etc. But I cannot mention everybody today. But this is work that has been carried out mostly by my student, Judith Ballesteros Villascan, and it's funded by the Newton Fund and CONACYT, the National Council for Science and Technology in Mexico. So we survey a number of traits, and I just want to show them just that because we are also doing many correlations in the Mexican Biobank to try to identify variants that are related to these traits, including some susceptibility to some pathogens. However, what we are focusing on in my lab is long non-coding RNAs. So to explore the variation of these molecules in Mexicans, in modern Mexicans, we selected all individuals that had over 90% Native American ancestry. And we used the Phantom Cat catalog, which was actually created by the Phantom Group, in which Pierre Carnisi was part of, or a leader on. And we're using that to check any selection in different archaeological regions. So how do we do this? We do this using a thing called population branch statistic, which measure if an allele has an extreme frequency compared to two other populations. So this basically detects very recent natural selection. And this is equates to having high PBS values. And to do this, this is one example. So for example, for the central region of Mexico, you will be seeing graphs with the number of PBS and compared to the number of SNPs we see in each long non-coding RNA. And for this, we compare one region to all the other different regions. So the in-group changes in each case against the Gulf of Mexico, Mayan region, North of Mexico, Oaxaca, Western Mexico, etc. And as an outgroup, we use the Han Chinese population, which is because the migration into of the populating into the Americas came from Asia. So what we find is there are a number of overrepresented alleles, and I don't have time to talk through all of them, but we see higher PVS values in the central and the Mayan region of Mexico. So we are definitely looking into those. And just to give you a taste of what we're finding, we find several variants in long non-coding RNAs that have selection signatures, including, for example, MIR22HC in the Mayan population, which is a molecule that long non-coding RNA that is known to promote regeneration in muscle injury. So with this, I just want to thank you for your time. This is where my lab is located at Langevio, and I think you should all definitely visit Mexico and Langevio. And I want to thank my lab, and this is a more recent picture, including Judith, who did most of the work I presented today. Thank you.
Thank you very much, Selene. Thank you very much for this also very interesting presentation. We've come to the end of this session. I'm really impressed by
I think we lost Maria.
We lost you, Maria Grazia. Maria Grazia, we don't hear you.
I suggest, Emilia, that we move on. I think obviously we are all very impressed by the talks that we heard by the institutes and by what you're all doing. And obviously some of you are much closer, as in the case of Selene, to Human Technopole and what's going on there than others. But obviously everyone has something interesting that they've presented, and their institutes look really worthwhile places to both visit and talk to. So
yes, I agree. We are half an hour late, and before moving on to the final session, I want to thank Martin and Roberto, who helped us on the technical level. All the logistic of the transmission was made possible by them. And some comments from the chat in YouTube. There are people connected from Funsalud, from Sinvestav Mérida, from Departamento de Biología Celular, perdón, I was speaking Spanish. People are asking if it will be possible to collaborate in cancer research or in microbiota. And Rafaela Schiavon makes two comments, one about gender approach to medicine and to genomics, and the second one about bioethical approach. So I think we can move to the final session. We have two new entries in this session. Dr. Guillermo Elizondo Suela, Head of Department of Cell Biology in Zacatenco unit of Sinvestav, and Dr. Juan Miguel Pascale, who is Director General of Instituto Gorgas in Panama. We don't have much time, but I would like to listen from you, maybe first from Jan and Alessandro, their impression about what they heard, the future potential collaborations, and then the same comments I would like to receive from the other people.
So perhaps, shall I start, Alessandro? Okay, so I think what we've heard and what we've seen is that there is an impressive array of research going on, different research institutes, individual institutes or organizations that are spread throughout the countries. I think really, however, that to talk about collaboration, as I said, we want to be collaborative, we want to be open, but to think about collaboration, my suggestion is that what's really needed is to encourage individual scientists to get into contact with people who they're interested with. interested by. One very obvious example was the last speaker. Selene is working in an area where, as she mentioned a couple of times, Piero Carnici, who is one of the heads of Research Center at Human Technopole, has been working for some time and is trying to continue to create resources. I think in order to produce and proceed with the possibility of collaboration, it's necessary for these people to get together and to talk to one another. Collaboration is not something that works top down. It's something that works bottom up, when people who are interested by each other's work get in contact with each other. So I think my encouragement would be to ask the heads of institutes to encourage their scientists to look at what's going on in Human Technopole, to get in touch with us. That can either be done at the level of Maria Grazia, who has rejoined us again, and you can see her email address in the chat, or it can be done by contacting the individual researchers themselves, if you prefer that. And I would simply encourage people to do this. I think it's really the only way to get from a high-level description of what's going on in an institute to actually working together.
Alessandro, would you add anything?
Yeah, no, I agree. I think it's very difficult to have, you know, bilateral collaboration that comes from top down. It's basically it's the research itself that puts you in close contact with your colleagues. It's research and infrastructure. So I just invited Selene to join. We are gonna have we're gonna have the inauguration of our EM facilities, just a celebratory. But of course, because of her interest in RNA, etc., I'm working myself with the machinery that transcribes DNA into RNA, etc. So she was curious to know whether somebody is gonna be interested to look at RNA from structural biology or biochemistry perspective, and that's certainly the case. And I think these events are very important exactly for the reason, because you give visibility to institutes which are far apart geographically and even time-wise. We work on probably very different, and getting to know each other is what fostered this. I mean, I was super impressed also. I thought that we had large fermenters, and then I've discovered that it's nothing compared to what you guys are setting up for other purposes. So I think it's fantastic that there is someone with the vim and power to organize this type of meetings, and I think it's great.
Yes, I agree with you. The idea of these events is to introduce these laboratories to the colleagues and let them know that these facilities exist, and then they can work together. But meanwhile, from an embassy, I am a researcher myself, but from an embassy, and from my experience, I also have to add that sometimes people start collaborating, but now to go on, they need something signed. So we have to sign an MOU or something. But I think that at first, the bottom-up approach is the most useful and convenient. I would like to give the microphone just 1 second to Maria Grazia, who wants to apologize with us. And
yes, thank you very much, Emilia. I'm very sorry, somehow my connection fell just while I was wrapping up the session and saying goodbye. So I just wanted to get a chance to thank the organizers again for putting this event together, for inviting Human Technopole and me, including me, and just to say goodbye and to exchange the invitation, meaning that if any of the speakers or researchers from their institutes happen to be in Europe or in Italy, do reach out and come to visit us in Human Technopole, and then we can deepen some of the conversations in person, which is always a nice thing to do. So that's it. Thank you very much.
Many thanks, Maria Grazia, and many thanks for your job as a chairperson. of this.
Yeah, and if I can just add one thing, that what Emilia said about the necessity for MOUs and stuff is absolutely correct. And on our side, Maria Grazia's team is the team that takes care of working out these agreements, generating the agreements, etc. So again, that's another reason to contact Maria Grazia.
Many thanks. It's quite important, but what is important is the real interest among researchers. No? They want to collaborate, and so as a second step, we have to think about the MOU. And I would like to know first, to hear first the comments of the new entries, Dr. Elizondo or Dr. Pascale.
Hi there, I'm Guillermo. I have a few comments. I'm going to try to be short. I think that it's very important to establish strategies that allows our institutions to maximize the potential collaborations. So more than specific projects, I would like to share some ideas that may help to strengthen future collaboration. The first one is, I think that maybe a good strategy will be to establish a cooperation between our institution through our postgraduate research programs. Actually, CIMBESTAB has around 65, 66 programs with more than 3,000 students. And several of these programs has already established with more than 70 international academic institutions, cooperation agreements with a co-direction figure. That is, a student who developed his research work in two institutions and under the direction of two researchers, get a graduation title from both institutions. We can also create new interinstitutional postgraduate programs. For instance, I think about maybe a bioinformatics postgraduate program that I believe CIMBESTAB needed. And in particular, talking about this program should not be too difficult to initiate, taking in consideration the communications technologies that we have now. And I think it's also very important to identify researchers to create multidisciplinary groups to study and develop early biomarkers and therapies for chronic and degenerative diseases. And in order to delineate potential collaborative groups, maybe we will have a series of symposiums or seminars with specific themes, maybe related to a particular pathologies, where interesting researchers expose their investigation lines. And through these mechanisms, start the conformation of these groups, these collaborative groups. And well, that are my ideas to try to initiate future collaborations.
Dr. Pascale.
First of all, thank you, Emilia, for organizing this spectacular meeting. I'm so happy that you were able to do it. And also, I think it was very nice to know what you are doing from Mexico and also in Italy. We are beginning a good collaboration, a very fast, good, and expedited collaboration with Italy. One of our collaborators is going to be in September for a master degree in Calabria. And in addition, I will be traveling to Rome because we are going to sign an agreement with the Istituto Superiore di Sanità. In other words, I think it's at the beginning of a very fruitful collaboration in the future. But I think the best way to do it is we need to define areas of collaboration in our institutions. So with Mayrin Solis, I will ask her to begin contact with the people working with the stem cells, for example, in at the Technopole. Yeah, and also we have a very strong group working in SARS-CoV-2. That will be another way of collaboration. And I think that the beginning would be begin communication between scientists, and as soon as we have something we can do together, we move forward to the MOU and continue with our collaboration. But anyway, I think it's at the beginning. Now we begin to know each other, and we know what we can do together and how we can maximize research to do something productive. So thank you very much for this organization, and for sure it will be not the last meeting that we have together.
Yes, I hope it is the first of a long series of meetings and opportunity of discussing these issues. Brenda, Tonatiuh, Ricardo Leonard, Kieran, would you like to add some comments?
I think I can, yeah, I can briefly give some I think it has been a wonderful portrait of what this new institute is doing. And of course, it would be great to have, I wonder if we can have some sort of inventory of research interests and researchers, so that we can circulate among our scientists this information and also exchange the list of all the researchers and what they are doing from our side. I haven't done the homework. I don't know if that information is available online.
Yes, the information on who's there and which researchers are there, what their research programs are, is all available online. So that's accessible to anyone. That's
perfect. Again, I would like to thank you very much because of the wonderful session, and it's an eye opener of new possibilities. And I think what you are doing is great. It's just amazing. And we are going to circulate among our scientists this information so that they can start contacting their peers and both. Yeah.
Thank you.
And I want to remind that the video was recorded, and so those who were not present today can see it on the YouTube channel of IBT. Brenda and Tonatiuh, would you like to add anything?
Yes. Hi, Emilia. Well, here at our institute, the way we have to sort of quantify the international collaboration is by tracking the publications we have with different countries. Actually, we have the last record we have is that we have the collaboration based on papers published together. So the last data we have is that we have at least 70 countries throughout the world with whom we share common publications. Of course, the United States is our largest partner in this sense. And the reason is simple, it's the closeness in terms of geography. And as you know, the problem we have in Mexico in the past few years is that financing has dropped dramatically. And this has difficulted the travel to Europe. When we look at the relation we have with Europe, we do have some main partners, which is Germany, Spain, France, and United Kingdom. Those sort of are our main partners in terms of common publications. And Italy is a little bit lower in that list. And I think from what we heard today, we have a tremendous potential to bring this relation to a higher level. And referring to the economic problem, what we have learned now today and during the past 2 years is that now we're closer than ever thanks to COVID. So we could, before, to bring some lecturer to Mexico, it would cost a lot of money, the airfare, stay, etc., and vice versa. Of course, there's nothing better than the personal contact, but with Zoom, with this new electronic ways of communicating, we could set up a program where we could establish a much more intensive exchange of seminars. With Panama, unfortunately, we don't, at least the record I have, we don't have common publications, but from what I've heard today, it's incredible, the development, the work that is being done there. It's beautiful, the knowledge city, and I think we do have very common interests. At the end, as Yain mentioned, things don't work when it's top to bottom. It has to be grassroots. And the way it has worked with us is we have tracked the collaboration to those cases where a professor spends a sabbatical in the country, or one of the students spends some 6 months in another country. And that's the best way to really start a fruitful collaboration. So in concrete, to conclude, because we need to exploit better this a more intensive program of share seminars, so we can identify potential collaborations, and then try to search for funds to enable this traveling scientists or students to get the ball rolling.
Yes, thank you for the comments. I agree with you. There is not a big collaboration with Italy. We are trying to increase the opportunities of collaboration. It's quite difficult at present, you know better than I. And I agree that we should organize more specific technical seminars. You should identify the subjects, and then we could build around this subject more technical seminars, so that you can start collaborating, and then the money, and then we can find the money. If there is a real interest, I think that we can find the money. Any
Brenda is waiting, I think. Yeah.
Brenda.
Thank you very much for the information. I would like to make some thoughts about what we had, what we lost, and what we have to recover soon. The first is, in spite, we haven't lost any international agreement from the federal government, from CONACYT with other governments. We still have more than 220 agreements valid and active. There's not a single bilateral exchange agreement active nowadays. And it's critical for us to have support, economic support for exchanges, both of students for studies and also of researchers to do specific projects. And also scholarships for Mexican students to go abroad to study have dropped clearly, especially in the renewable energies field, from 1,100 to less than 200 a year. So we still need to give more scholarships for students to go abroad to study, but mainly for us as researchers, we need to recover the money for bilateral exchange stays. So now we are clearly based only on institutional funds for that, but it's not enough. We need to make a claim for those programs, those budget programs, to go back and to help us. Otherwise, it's going to be very difficult to keep these international relationships growing. Perhaps our generation, who has several years working on different countries with different collaborators, we can extend for a while, but young researchers need to move to make some content, to make specific collaborations to last for a career. So it was very useful resource for us. Now we lost it, but I want to say that there's something we need to recover as soon as possible. Thank you.
Thank you, Brenda. About the comments in the YouTube channel, some people were asking about collaboration in cancer research and in microbiota. I don't know the English for microbiota.
So at this point, we don't have anyone who's doing microbiome research. That's something that we would like to establish, but given the current focus on setting up some national platforms, I'm not sure at all whether we can confidently say that this will happen or not. There are a number of researchers who are interested in cancer from different aspects, mainly, I would say, from the bioinformatic and computational aspect. And the person who is the head of the Computational Biology Research Center, Andrea Sotoriva, is one of those. Another is one of the group leaders in that center, Francesco Iorio. But there are a number of researchers who have a mix of activities, some of which are related to cancer research and some of which are not. I think one of the earlier comments that came up was about rare diseases. And Nicole Soranzo, who is in charge of the human genetics and genomics area of the Genomics Research Center, is very interested in the genetics of rare diseases. And so there are clear interfaces for some of the areas that have been mentioned. And I would again encourage people to find out more details about what's going on in Technopole and simply contact the people, or via Maria Grazia, contact Human Technopole, and Maria Grazia's team, or Maria Grazia herself, I'm sure, will pass on your message to the most appropriate people.
Thank you, and I can also help. So if they want to contact me, I can
absolutely
to reach the right person in Human Technopole. And there are also people from the Instituto Politécnico Nacional who are assisting to this webinar. Welcome and good afternoon. Yeah, it's afternoon here now. And I don't know if there are any more comments from you, because if not, we can conclude. Tonatiuh.
Yeah, just a comment about the areas of cancer and microbiota. In our institute, there are several people working on developmental biology, particularly using as experimental models zebrafish and Drosophila. And the work is, it's basic research, but has many interesting implications in terms of cancer research. And there, we also have a couple of important groups working on the genomic analysis of indigenous population in Mexico, and they're obviously they're generating a tremendous amount of information that can be of interest for cancer, and also for neglected diseases, particularly that affect part of our indigenous population. And in the microbiota, we have also a couple of groups. There's a very interesting group that has done an extensive mapping of the waters in the Gulf of Mexico, different from the surface down to the bottom. And now they have a tremendous collection of samples and have been characterizing the microbia, all the microbiota in this samples. And also some groups working on the microbiota of gut of shrimp. That's important for fisheries, and in the mouth of patients and gut of humans. So there could be also some interesting work that can complement our capacities with our colleagues from Italy and Panama.
Thank you, Tonatiuh. And about fundings, I want to stress that the embassy is doing tremendous efforts to find a way to enrich the opportunities of collaboration, bilateral collaboration in science between Mexico and Italy, and in the future also between Panama and Italy. We have just signed a new agreement with the government of the state of Guanajuato. It is on agriculture, it's not on life sciences. And now we hopefully will sign the new protocol between the two Ministry of Foreign Affairs with an addendum dedicated to health sciences. It's quite difficult, but we are doing our best to increase these opportunities of collaboration. Before moving to the closing session with my colleague Giulio Mignacca from Panama, I want to thank all the panelists, all the people who made this webinar possible. I want to thank people from Human Technopole and in particular the Director General. I wait for them here. I hope we can organize a real meeting in Mexico, in Panama, in both countries, before I leave this place. I will be here until February the 23rd, 2023.
Yeah.
So I hope it will be possible to meet us here.
So thank you very much. And from our side, thank you very much for organizing this and to everyone for participating. It's been very interesting for us.
Many thanks. And so, Giulio, if you are here, you can give your...
Yes, Emilia, thank you. Thank you again. Thank you, everybody. As I always say, the best thing about being a diplomat is that every day you can learn different things, different stuff, and then you can move from cultural issues to scientific issues. So thank you, because it was very, very interesting for me as well. And let me thank you on behalf of Ambassador Nicoletti. He's out of country for official mission, so couldn't join us, but he was very, very happy of this event. It was Ambassador Nicoletti that asked the extension of the competencies of Emilia Giorgetti to Panama as well, because he firmly believes the importance of scientific diplomacy, scientific cooperation. So at the moment in Latin American region, we have scientific attaché in Mexico, Brazil, Argentina, maybe in Chile, but then Panama is the only other country of this list. So it's a very it's a privilege for us, and let me thank you again, Emilia Giorgetti, for organizing this webinar, and also to create these connections between researchers. I think Director Alessandro Vannini was mentioning the importance of these exchanges, between researchers at all levels. And so we as government are trying to push the scientific cooperation, but as you speak the same language, it's of course easier for you. We know the great friendship and cooperation existing between Mexico and Panama, of course. It's very common with Emilia. We saw that when we met, when Emilia visited us in Panama, we arranged for her a large series of meetings with Senacid, with Instituto Gorgas, with all the ministries, environment, etc., and Smithsonian Institute. And it was very, very common to hear that scientific from Panama, they had an experience, an academic experience in Mexico. So we are now pushing this and also presenting our excellencies from Italy. Human Technopole for sure is one of our excellencies. It's a very recent organization, but working on a very strategic field, scientific field for the future, as life sciences, human sciences are. The pandemic showed us the importance of cooperation between researcher and academics. And I think that the worldwide efforts through a cure for the COVID or to the development of the vaccine helped us to not to finish with the pandemic, but for sure to move forward to new stage we are. And so we already started here in Panama this process to create connections with scientific institutions. We already have a very, very good cooperation both with Instituto Gorgas and Senacit. And INDICASAT, as you know, it's also controlled in some way from Senacit. So our relation with Dr. Ortega, it's very, very interesting. He actually had an experience in Italy as well. He worked in Siena. So this is how I think scientific relations work, with interchange, with travels, with missions, and with this very positive cooperation. As well with Instituto Gorgas, Dr. Pascale, we also awarded, the President of Republic of Italy awarded him with a decoration last year for his impressive job during the last decades as a scientist here in Panama, and also as an descendant of Italian immigrants here in the country. So the embassy is here, as well as the Embassy of Italy in Mexico. We work to create connection. We work to help find money sometimes, because you know how money is important for research, and so we can help in and find projects, bids, whatever is existing internationally. And we are here, and we really believe in this scientific diplomacy. This is the message from the Italian embassy in Panama, but also from the Ministry of Foreign Affairs and International Cooperation. So thank you all, and keep in touch.
Many thanks, Giulio. Many thanks to everybody. And let's plan the visit of Human Technopole in Central and North America.
No, nosotros queremos ir.
Goodbye.